Apoptosis, a form of programmed cell death, has been induced in these cultured cells. Cell death is characterized by blebbing of the plasma membrane and fragmentation of the nuclei. Suddenly, cells weaken attachment to the substratum that they have been growing on and shrivel up without lysing. In the following movies, we observe the process at higher magnification. The mechanism of apoptosis involves many tightly controlled steps, 3 of which are demonstrated here by different visualization techniques. One initial event is the sudden release of cytochrome C from mitochondria into the cytosol. This event has been visualized here using fluorescently labeled cytochrome C. Initially, the greenish-yellow staining is restricted to a reticular pattern, which then suddenly disperses as the mitochondria release their content proteins into the cytosol. At a later step, the lipid asymmetry of the plasma membrane breaks down. In normal cells, phosphatidyl serine is found only on the cytosolic face of the plasma membrane. But when cells undergo apoptosis, it becomes exposed on the outside of the cell. This event has been visualized here by adding a red fluorescent protein to the media, which specifically binds phosphatidyl serine head groups as they become exposed. In an intact organism, exposure of phosphatidyl serine on the cell surface labels the dead cell and its remnants so that they are rapidly consumed by other cells such as macrophages. Finally, although apoptosing cells don't lyse, their plasma membranes do become permeable to small molecules. This event has been visualized here by adding a dye to the media that fluoresces blue when it can enter cells and bind to DNA. All 3 of these events can be observed in the same group of cells.